Good afternoon and welcome to Penn Nursing's special Preceptor Recognition session. On behalf of our faculty and students, Penn Nursing extends its deepest appreciation to all of you who teach, advise, mentor, coach, and inspire our students every day. I'm Rosemary Palomano. I'm the Associate Dean for Practice. And please know that your partnership with us is truly exceptional, and your ability to provide amazing experiences for our students are truly valued. We had hoped to continue our longstanding tradition in providing a Preceptor Recognition event, which is an all-afternoon event, but unfortunately we decided to postpone it until early fall. And we will be sending a save the date out shortly. But we just couldn't wait to provide this current and timely program to you. It's my pleasure to introduce our Dean for welcoming remarks, Dean Antonia Vierwell.
Thank you, Rosemary, and welcome to this year's Preceptor Recognition event. We come together today to recognize and thank you, all of you, for precepting Penn Nursing students, for modeling nursing excellence, and for being the great teachers for our next generation of nursing leaders. Education and practice cross paths in the preceptor role, and we are incredibly grateful for your work over the last year. I know that the last 15 months have been particularly challenging for you and all nurses. The fact that we are meeting remotely today speaks to the challenges all of us face as the pandemic begins to ease. This pandemic has made it a tough year on so many levels, and it continues to cast a shadow on your work. Or I should say, it continues to shine a light on your work, because your decision to precept students, even under these circumstances, makes you a true champion for the future of health and healthcare. Your enthusiasm, expertise, and knowledge are valuable, and I want you to know how appreciative we are. Myself, our faculty, our students. We know how hard you work. We know how much energy and effort you put into precepting. We see you. Thank you barely seems adequate, but I want to repeat that and say that again to each of you. Thank you. Penn Nursing is the #1 ranked school of nursing in the world for the 6th consecutive year. It's true that this ranking is reflective of our students and our alumni. The world recognizes the leadership and superior skills that our students and alumni have developed. But that can't happen. It wouldn't happen without you and your work to precept our students. It would be so much more difficult to prepare them for a future in nursing. You are an integral part of our educational experience. So thank you again. Thank you for your mentorship, your teaching, your feedback, your encouragement, and for being part of our Penn Nursing faculty. We appreciate your sharing your successes and experiences, but most of all, thank you for the work that you do. Every student you touch carries your knowledge and experiences with them. You are that voice in the ear that stays with them years after they graduate from Penn Nursing. A preceptor is special. You are special. Thank you for that you all do for Penn Nursing and for our students. Again, I know you're going to enjoy this event, as will I. Take care and look forward to seeing you in Fagan Hall soon.
Thank you. So I just want to go over a few details for the program today. We are offering one contact hour and only for those attending this live virtual event. A link will be posted in the chat area during the presentation, and at the end of the presentation, you will actually go to that link. You will complete an evaluation, submit the evaluation, and you should expect to receive your certificate in about 2 to 3 weeks. We do want you to, this is an exciting program, we do want you to ask questions. If you have questions during the presentation, please place them in the chat area. I will be monitoring those questions and then presenting them to our speaker. So it is my distinct honor to introduce our speaker today, Deborah Dunbar, who is Clinic Coordinator for the Penn Prevention Research Division at the University of Pennsylvania Perelman School of Medicine. Deborah has had an impressive leadership career in clinical research. She has been a leader, a manager, and a director of clinical research units, supervised and managed all levels of research staff and teams, and provided oversight for large research budgets in the millions of dollars. She is currently part of Penn Medicine's team responsible for launching the landmark Moderna COVID-19 vaccine multi-site randomized placebo-controlled clinical trial. She spearheaded and orchestrated one of our Penn Medicine's largest effort in urgently recruiting, onboarding, training, and supervising research staff, putting together and managing, again, a huge research budget, and expanding Penn Medicine's data collection sites in response to a global pandemic. We are proud to have Deb here today with us to present an update on the research for COVID-19 vaccines. And she is going to clarify and amplify the accurate information around this research and how it translates into practice, counseling of our patients and the public, and understanding what's to come with this global pandemic. And we are especially proud because she is one of our alum, having received her MSN from Penn Nursing in adult primary care and her BSN at St. Anselm College, graduating cum laude. And so I am in awe of her knowledge of vaccine science, her execution of clinical trials, her ability to translate research to guide clinical decision-making around this current pandemic. So I know that you are going to find this session very informative. So please join me in welcoming Deborah Dunbar.
Thank you so much, Rosemary, for that incredibly kind introduction. I can just say that I was in either the right spot or the wrong spot at the right time, depending on how you look at it, when the pandemic began. And, that ended up being how things went for me. Can somebody tell me, are you able to see my slide?
Yes.
Yeah. All right.
Okay, super. So let's not waste any time. There's a lot of information I have to share with you. Very briefly, these are the topics that I'd like to discuss over the next 45 minutes or so. And I think that they're the critical topics for all of us to be familiar with as we counsel patients, colleagues, on what are really the facts related to COVID vaccines. There's a lot of misinformation, as we all know, that's circulating. And so what I hope to do today is leave you with lots of information based in science that you can take out to your communities and to your practices. I think the SARS-CoV-2 pandemic really needs no introduction. As we all know, the SARS-CoV-2 virus was actually identified back in December 2019 in Wuhan, China. A little bit unclear at this time exactly what the source of that virus entering human population is. And I think there's more information that we'll be learning about that. SARS-CoV-2 stands for severe acute respiratory syndrome, and then coronavirus 2. There was a SARS-CoV-1, as you might imagine. This was originally called the novel SARS-CoV-2 SARS-CoV virus, and then eventually it was settled upon this for a name. When you hear COVID-19 referred to, what that stands for is coronavirus disease, originating in 2019. So they're really not the same, although we do hear them used quite interchangeably. I want to just quickly orient you to a single virus particle, or a virion of SARS-CoV-2, because we're going to talk a lot about these spike proteins. The spike proteins exist on the surface of the virion, and they are the target of all of the vaccines that are being investigated or have EUA authorization at this point in time. They make a really nice target because they do exist on the surface, and they're also the part of the virion that latches onto the receptor on the human cell. So if we can come up with a vaccine that blocks these spike proteins, that's a really important step forward in managing our pandemic. I also want to talk with you a little bit about how vaccines are developed in general, not just the COVID vaccines. And you can see here that most vaccines take some time to develop, from the time of the discovery of the virus to the vaccine that's actually developed and marketed for human use. So influenza vaccine was remarkably short. Not listed on this slide is the Zika vaccine, which also was done very rapidly in under a year. You can see we got a lot of questions about HIV vaccine and why that's not yet developed, but you can see, despite the fact that we've been working on that for almost 40 years, that has not yet happened, but that's not actually that unusual. And of course, I know everybody on this call is familiar with the fact that vaccines have really changed the landscape of healthcare and really are very responsible for extended life expectancies. The CDC was asked to list the 10 most important health developments in the last century, and number one by far and away was the development of effective vaccines. And so it's no surprise that a COVID vaccine would be very important for managing our pandemic. So all vaccines begin pretty much the same way. There's a problem, like a pandemic or an epidemic. Someone has an idea about how to actually manage or develop a vaccine against that particular pathogen. Lots of lab research happens, and that can be sometimes the longest phase of vaccine development. And once it appears that there's a candidate vaccine that has at least preclinical data that supports taking that into moving that along in the process, it moves to animals. And typically animal studies begin with smaller animals and progress to larger animals, almost always ending with chimpanzees or monkeys, because their immune system very closely resembles our own. Once we have demonstrated safety and a good efficacy signal in animals, it moves to human research. Once we get to human research or clinical trials, there's basically 3 phases. The first phase is typically a very small group of healthy people, and it's quick, and those studies are done to determine whether or not this product is safe. Once it's determined to be safe in a small group of people, we'll move to phase 2 research. That lasts a little bit longer. We enroll additional subjects, and we begin to study not only safety, but whether or not there's an immune response. And if both of those things are demonstrated, it moves to phase 3. Phase 3 studies involve enrolling large numbers of participants who are at risk of the disease of interest, and they last much longer. When I talk about, one of the things that I think people are probably thinking about is, well, how did we possibly move through these stages so rapidly in developing a COVID vaccine? And this is just a nice little schematic, again, to show you the various steps, and then, of course, the post-study steps of licensing, manufacturing, distribution. When we look at the COVID-19 vaccine development timeline, it was really compressed to much less than a year. And so let me explain to you a little bit how that happened without skipping any steps, without sacrificing safety, and with knowing that these vaccines are not only safe, but incredibly effective. So the preclinical phase, I can tell you much of that work was done before the SARS-CoV-2 virus was actually discovered in that December of 2019. This virion resembles, as I said, the SARS-CoV-1, and it also resembles several other respiratory syndrome viruses like MERS. And coronaviruses, MERS, those kinds of viruses had been very extensively studied prior to the onset of this pandemic. So there was lots of data and lots of thinking that went into this before we even got to that point in time. And then instead of running the human clinical trials sequentially, we actually compressed this and ran them with some overlap. So once we had some demonstrated safety in phase 1, we moved to phase 2. Once we had additional safety data and some efficacy data in phase 2, we went on to phase 3 while those studies completed. And during the phase 3 studies, because of Operation Warp Speed, manufacturing was begun. So that's a financially risky thing to do. We didn't know at that time that these vaccines would be going to market, yet the power and resources of the US government were put behind manufacturing so that at the moment that it was determined that these were safe and efficacious, they could get EUA and be distributed and then injected into human arms. And so there were no steps that were skipped, and there's ample evidence that these are safe and efficacious vaccines. So just to talk for a moment about what EUA means, because I think there's a fair amount of confusion. Emergency use authorization is a mechanism that the FDA has put in place for dealing with things like a public health emergency, exactly like the COVID-19 pandemic that we've had. I can tell you I've never seen an EUA authorized vaccine before, and I've been doing this for a little bit. But this is exactly the kind of situation where it's great that we have this mechanism. The standard for getting EUA authorization for a vaccine is the FDA has to determine that the known and potential benefits outweigh the known and potential risks of the vaccine. It's a slightly lower standard than we have for full FDA approval, but also a really important standard and one that was met and exceeded for the 3 vaccines that do have EUA authorization. There's also something called post-marketing surveillance or phase 4 research. I've seen this in some HIV medications. These are clinical trials that happen after the drugs received full FDA approval, but perhaps the FDA or the pharmaceutical company who manufactures the vaccine wants to gather just a little more safety or efficacy data, and so post-marketing surveillance happens. You're all familiar with full FDA approval. And then I also just wanted to mention the role of the Advisory Council on Immunization Practices, or ASIP. That's a group that operates under the Centers for Disease Control, and they make recommendations about what vaccines should be administered on what schedules. And I'm sure everybody here is very familiar with those charts that we use for administering vaccines in our practices. Vaccine mandates are, of course, a fairly controversial topic, although it's not a new controversy. And I want to just mention a little bit around this. Many people on this call may be familiar that the University of Pennsylvania, and as well as the University of Pennsylvania Health System, has now opted to mandate COVID vaccination for all staff, faculty, and students. They've set forth a process, and they've set forth some exemptions. And the university is not alone in this. There are other universities and other health systems, as well as other employers who are doing this. There is a common myth right now that vaccines cannot be mandated if they only have EUA authorization. That's really very legally murky territory. There is no law that says you cannot mandate an EUA vaccination. There's also no law that says you can. And so I suspect we will see cases make it to the courts, and that will be carefully watched. And then, of course, there's issues that we need to be thinking about before they happen. What if somebody who really didn't want to receive a vaccine has a serious adverse event, perceives that adverse event to be related to the vaccine, and they only got the vaccine because it was a mandated vaccine? What will the legal ramifications of that be? And then, of course, there's also privacy issues. What if vaccines are mandated, and those who don't receive them for perhaps a medical condition or a religious belief that they'd like to keep private from coworkers, perhaps they have to wear masks in their work setting because of those exemptions. And is that going to communicate something that they wish wasn't communicated to their coworkers? And so all of these things are really going to need to be worked out. All right, let's talk about the vaccines themselves. The vaccine designs that most of us are most familiar with are the two that appear with X's at the top of this slide: whole inactivated vaccines and live attenuated vaccines. So oral polio vaccine is a really good example of a live attenuated vaccine. And many of us know that you can get a very mild case of polio in kids who get oral polio vaccine, the oral version. because we don't really want to take any risks for people getting a mild case of COVID-19, those strategies are not being used at this time for development of COVID-19 vaccines. And I'll call your attention to the bottom left hand of the screen, because these are the two strategies that are being used in the EUA authorized vaccines in the US right now. The first one is using recombinant viral vectors. That's used by AstraZeneca and Janssen. Janssen is a subsidiary of Johnson & Johnson, so you may hear it referred to in that way as well. And then the other strategy that I'm sure you're all familiar with because it's received lots of exciting press is the strategy of using messenger RNA to deliver instructions. That's the strategy that's been used by both Moderna and Pfizer in development of their vaccines. So let's just talk for a moment about how an mRNA vaccine works. In both cases, Moderna and Pfizer, they are dosed, there's 2 doses that are given in order to achieve full immunity. So if you follow the graph on the right, the first step is the mRNA is encased in these lipid nanoparticles, and then it's injected typically into the deltoid, to the non-dominant deltoid. Once it gets inside a muscle cell, the lipid nanoparticle then unsheaths the messenger RNA. That messenger RNA in the cell then provides instructions to assemble amino acids into proteins, and the specific protein is the SARS-CoV-2 spike protein. Those spike proteins, once assembled, then sit on the surface of the muscle cell where they're recognized by our immune system as invaders or foreign. And our immune system, our B cells, then activate an antibody response, and as well as a T cell response, CD4 and CD8 cell response. And so then we have developed an immunity to the spike protein so that if in the future we encounter a spike protein on the surface of a COVID virus, then we're prepared and we have circulating antibodies. One of the reasons a lot of people say that they don't want to receive the mRNA vaccines is there's this perception that somehow it's going to change our DNA. The truth is that's not possible. mRNA is a signal to the cell, and it only goes to the cytoplasm of the cell. It never enters the nucleus where the human DNA is located. The mRNA is also highly degradable. Once it provides instructions for this assembly of spike proteins, it degrades and the immune system's been primed and the mRNA is gone. And mRNA vaccines have been, again, there's lots of research that happened prior to the onset of this pandemic. So there's lots known about this. And so while they're new in widespread application, they are not new in much investigation. All right, let's talk a little bit about how viral vector vaccines work. And it's actually quite similar to what I just explained, except for we back it up one step. Instead of encasing the mRNA in lipid nanoparticles, what happens here is DNA coding for spike proteins is encased in a vector. You can think of a vector as an envelope. If I write you a letter and I drop it in the mailbox without an envelope, it gets nowhere. If I write you a letter and put it in the envelope, that works much better. The vector is like that envelope. It gets the vaccine, the insert where it needs to be, and then the envelope or the vector becomes irrelevant. Adenoviruses are commonly used as vaccine vectors. In the case of the Johnson & Johnson vaccine, they use a recombinant adenovirus 26. Adenovirus causes the common cold. AstraZeneca uses a chimp adenovirus, and AstraZeneca, I should mention, has been investigated in the U.S. It's the most distributed and the most given vaccine on the globe. However, it does not have U.S. approval. All right, this is a little metaphor I use. For some reason, what people remember from their high school biology class is that a vaccine contains a little dose of the pathogen of interest. And it is understandably difficult to convince people to get a vaccine if they think they're going to get a little dose of what has turned out to be a very serious virus and pandemic. The way I try to explain this, and it seems to be understood by people, at all levels of cognitive ability, If I'm walking up the street and I see a bicycle tire and a set of handlebars, I immediately recognize that that's a bicycle. Yet I cannot put those two things together and ride it up the street. It just can't do what a bike does. It's not all the parts. If I administer a vaccine that has messenger RNA that then assembles spike proteins, and those spike proteins are circulating and cause an immune response, I hope that my body is going to recognize that immune response, those antibodies, against the spike proteins and fight off the virus. But those spike proteins resulting from the mRNA are not a whole virion, and they can't do what the virus does. So it is absolutely not possible to get SARS-CoV-2 or COVID-19 illness from these vaccines. Recall, they're not whole inactivated vaccinations. Okay, I'm not going to read this all to you, but I would like to go through each of the vaccines that does have EUA authorization so that you have a sense of what's out there and what the highlights, what sort of makes each one a little different from the others. So the Pfizer vaccine, the adult study, which enrolled folks who were 16 and older, enrolled over 43,000 people, and it was the first study to launch. So that was back midsummer, July through November. The other thing I want to say about this study, and it applies to the other studies that I'm going to describe to you, it much more closely, the populations they enrolled much more closely mimicked real world populations than most phase 3 studies do. And the reason for that is, of course, these vaccines are really important to be safe and effective in people who have serious comorbidities. It was very important that we discover that along with everything else. So rather than excluding people like we usually do in a phase 3 study, where it doesn't really resemble exactly the population that most needs to get the vaccine, we enrolled people with serious heart disease, diabetes, morbid obesity, the things that real older people. We have a fellow who's almost 90 who enrolled in a study in the Moderna study at our site. So we really did include real world populations. In each of the studies that I'll describe, recipients were randomized 1-to-1, vaccine to placebo. And then for Pfizer, it's 2 doses, 21 days apart. And you can see here that the efficacy was just amazing. As one who conducts these studies often and looks at results, I have never seen as compelling a result, as unequivocal a result as this. So at least 7 days after the second injection, when we assumed somebody would have full immunity or close to full immunity from the vaccine, there were 8 COVID-19 cases in the vaccine group and 162 in the placebo group. And if you look at severe cases, there was only 1 in the vaccine group and 9 in the placebo group. The vaccine was very well tolerated, and most of the reactogenicity type symptoms resolved quite quickly. There were no serious adverse events that exceeded the expected background rate, and the serious adverse event rate were very similar in both the vaccine and the placebo groups. So this was the first study. And just a note about endpoints. When we do our HIV vaccine studies and most other vaccines, it's not in the midst of a pandemic. So it does take that 3 to 4 years to develop, to collect enough endpoints to say something about the efficacy of a vaccine. When you do a study in the midst of a pandemic where the people who are enrolled are at risk, you can collect enough endpoints to have the power to say that the statistical power to say that there's a difference, that the vaccine makes a difference in a very short period of time. So that can happen in 3 or 4 months, like we saw in these vaccines. Pfizer also conducted an adolescent study. Some of you may have heard that they have received emergency use authorization for children ages 12 to 14. And the vaccine was even more efficacious in kids, likely because their immune systems are still young and incredibly active and able to mount, a really nice response to the antigenic stimulation of the vaccine. So this was the first vaccine also to receive EUA for kids 12 to 15. They have a study of children between 6 months and 11 years underway, and I would expect before the end of the summer we'll hear about that as well. All right, Moderna, quite similar. This is a study we did here at Penn Medicine. Moderna enrolled 30,000 subjects, a little more, They did not enroll 16 and 17-year-olds, but started at 18, and Moderna enrolled with 2 doses delivered 28 days apart. You can see here again, we have these amazing results. 11 infections in the vaccine group, 185 in the placebo group, and 0 severe cases in the vaccinated group, 30, including a death in the placebo group. So once again, highly efficacious. It was also found to be very safe. Most of the reactogenicity symptoms were mild or moderate and only lasted a few days. I do want to call to your attention, there's an unusual reaction that's been observed in a small number of people who received Moderna, and that's a delayed injection site reaction. It occurs typically starting at day 8 or 9, and at the injection site, it's a fairly large erythematous macular area. It's red, it's warm to the touch, and typically it resolves over the course of 2 to 3 days. It does not require any treatment. You know, people can take over-the-counter analgesics if they're uncomfortable, but that's unusual. We saw it with one of our investigational HIV vaccines as well. All right, Moderna also did an adolescent study, in this case 12 to 17 people. Once again, very efficacious, 100% so. After the second dose, there were no COVID cases in fully vaccinated adolescents. This is not yet published data. I took the information I have here. This is from a press release, and I think this was just last Friday that this came out. All right, let's talk about Johnson & Johnson a little bit. So this again is an adenovirus vectored vaccine. The adenovirus used in these studies are replication incompetent, so they cannot actually cause the cold that they might otherwise cause. They really are just that delivery mechanism. Janssen enrolled 44,000 plus subjects, a little bit later and a little more broadly around the globe than other studies. And Janssen has the advantage of being a single dose vaccine, resulting in full immunity. This is nice for patients who you're not quite sure are going to come back to the clinic, or you may track down again at the right period of time in order to administer the second dose. Once again, found to be safe. The most common reactogenicity symptoms here, injection site pain, just like the others, and then some headache and fatigue. So I know this slide is a little fuzzy. I'm showing you the efficacy data for the Janssen vaccine. This is the data collected around the globe, not just the U.S. The numbers are much better in the U.S., where there are fewer variants circulating, and they're more in the 85% range with regard to efficacy. So while a little bit less efficacious than the 2 mRNA vaccines, still really exceeding what many vaccines do. So when you're considering these various studies, I want you to remember they're all really good, they're very safe, and they're very effective vaccines. I saw a post this morning out of CHOPS Vaccine Center, Dr. Paul Offit, who is really, a lifelong vaccine scientist. He said very clearly, these are amongst, or even more so, the safest, most effective vaccines we've ever created. So we're in quite a fortunate position as far as that goes. The other thing I talked about was the fact that these study populations really more closely resemble real-world populations. And actually, the data coming from people after the studies, people who received the vaccine, is even better than the data in the vaccine studies, largely because during the vaccine studies, we ran really close surveillance looking for infections, including asymptomatic infections. We swabbed people regularly. We probably don't pick up those asymptomatic infections in the community setting, only the more serious and symptomatic infections. So once you eliminate all those asymptomatic cases, the efficacy even looks better. And it's also really difficult to compare these vaccines to one another. Each one of the studies was done in a different place, in a different population, at different times when the circulating variants were more or less prevalent, and they operationalized their endpoints differently. So what Moderna called a severe case of COVID is different than what Johnson & Johnson called a severe case of COVID. So it's hard to compare these. I want to talk about 2 special safety issues that have received some public, some press, and just make sure you're familiar with what's happening. So there have been reports of myocarditis in some teenagers, more typically boys than girls, with an onset typically within 4 days of receiving mRNA vaccines. At this point in time, the CDC is gathering those cases through a mechanism called VAERS, or the Vaccine Adverse Experience Reporting System, and trying to begin to better understand whether or not these are actually associated with the vaccine. The cases have tended to be mild and self-limited. It is possible that these are purely by chance. If you think about the number of teenagers who are being vaccinated right now, once we got EUA of the Pfizer vaccine in that teen population, and soon to add Moderna to that, the numbers are enormous. And so the last I saw this was under 10 cases, but we should all pay attention to this. And then, of course, I think people are familiar that the Johnson & Johnson vaccine was on a temporary hold. There were some, I believe there were 7 or 8 cases, thromboembolic events in the study of people who received vaccine, but with the Johnson & Johnson vaccine, but they were thought to be not associated with the vaccine. The rate was quite similar with the placebo groups. However, with both AstraZeneca and Johnson & Johnson, and of course these are the two viral vectored vaccines, there have now been many cases of cerebral venous sinus thrombosis with thrombocytopenia after receiving an adenovirus vectored vaccine. This has been observed both in the US and in Europe. Lots of data is being collected right now. It's most typically observed in women who are under 50 years old and within 3 weeks of vaccination. Some of those women were on OCPs in a small case series that was reported. Some were not, and they still had this illness. The most common presenting symptom is headache, followed by nausea, vomiting, lethargy, fever. So those are good things to be aware of in the clinical setting. And their association with the vaccine is not clearly established, although as we collect more cases, it'll be interesting to see. It won't surprise me if a loose association is in fact identified. So I want everybody to just be aware that this all involves some complicated risk-benefit calculations. When we vaccinate millions of people, like we're doing right now with COVID-19 vaccines, life is going to continue to happen and some serious adverse events will occur. And adverse events are just illnesses, things that we wish didn't happen. It does not necessarily imply that they're associated with the vaccine, because we certainly record adverse events in placebo recipients as well. There needs to be, for each person, a really careful, individualized assessment. What's the risk of developing a serious, life-altering adverse event with the vaccination versus what's the risk of their morbidity and mortality associated with getting COVID-19? And to date, there is not a group that's been identified where the risk of the vaccine outweighs the risk of the disease in the midst of a pandemic, which is why these are so broadly recommended and considered to be so safe. And so that you understand how the association with the vaccine is assessed, you can see here, basically, there's lots of ways to look at this. Is there a tight temporal relationship? Did the person have a predisposing condition that may have led them to develop this particular adverse experience? So there's lots of very careful evaluation that happens in order to decide whether or not a particular adverse experience is related to the vaccine. So I want to run through quickly some special populations. And probably the most important one is, what do we do with people who have a history of having had COVID-19 illness? CDC recommendation right now is to wait out to 90 days. In practice, most of what is happening is to wait at least 30 days. And because people who have COVID-19 develop these amazing immune responses after one vaccine, it is possible that they don't need their second vaccine in the two that require a series of 2. In fact, the highest antibody levels that have been observed are in people who had COVID, waited 30 days, and got a single vaccination. And so it may not be necessary to do that second vaccine. We'll see what the CDC says down the road. Pregnant and lactating women, there's minimal data, but it does appear to be quite safe in pregnancy. Another common myth is that COVID vaccines have some impact on fertility, specifically reducing fertility. That has not been demonstrated. There's no data to support that statement. And there are antibodies against COVID that appear in breast milk and are passed to a nursing infant. We do not know if those antibodies confer protection in the infant. However, it's somewhat reassuring, I think, that the antibodies do go through the breast milk to the infant. As far as immunocompromised people are concerned, if it's at all possible to complete a vaccine series prior to it initiating any kind of medication that would be immunocompromising, that of course would be desirable. But in the many cases where that's not possible, it is absolutely safe to vaccinate people. There's demonstrated safety. We included people with well-controlled HIV in this study, so there's demonstrated safety in people who have HIV, people who are on cancer immunotherapies, and people who are post-transplant. I can tell you that solid organ transplant recipients and those who have hematologic malignancies do have very poor antibody responses, and their T-cell responses have not yet been well described. I heard a presentation last week where one strategy was to give a heterologous vaccine booster a third vaccine. So if in these cases of people with solid organ transplants, let's say they got 2 Moderna vaccines, wait a month and give a Janssen vaccine, and that actually did induce a nice response, but that's not published data or recommended anywhere at this point. Another interesting special population are people who have dermal fillers. These are fillers like Juvederm, things done for cosmetic reasons. After receiving mRNA vaccines, some of those people did find that the site of the filler became locally reddened and swollen. And again, it was quite self-limited. There were no safety concerns. It was more, again, a cosmetic issue. So that's a good thing to sort of let our patients know that may happen. It's safe to give vaccines to people who have a history of Guillain-Barré, people who have a history of Bell's palsy, and people with autoimmune disorders. For folks who have a history of anaphylaxis, as long as it was not precipitated by one of the ingredients in the vaccine, it's okay to vaccinate. What we typically do is observe them for a bit longer than the 15 minutes that's recommended. We keep them in the clinic for 30 minutes. People who have a genuine allergy to polyethylene glycol, or PEG, need to be carefully evaluated. So PEG is used in many vaccines, including the flu shot, including Miralax, non-vaccine products. So any history of safe receipt of a flu shot or use of Miralax really does rule out a genuine PEG allergy and would let you know it's okay to proceed. All right, let's talk for a moment about breakthrough infections. The CDC defines a breakthrough infection as somebody who developed a COVID-19 case at least 2 weeks after the final vaccine in whatever series they received. And you can see here that the numbers are very low. So there were a little more than 2,400 cases out of more than 130 million people who've been fully vaccinated as of the end of May. You can see the rates are just incredibly low, 0.0018% breakthrough infection rate in this particular set of data that's been collected by the CDC, and an even lower breakthrough death rate from people who've been vaccinated. And just note that breakthrough death rate, not all of those deaths were necessarily from COVID-related causes. So that number is actually even a little higher than exists in reality. So breakthrough infections do happen. The vaccines are not perfect. However, the numbers are incredibly low. All right, probably the thing that's interesting, or the most interesting to most of us, is this whole issue of variance. So remember that variants occur just through random mistakes in viral replication. If we can vaccinate everybody around the world, the virus has no opportunity to replicate, reproduce, and so those variant mistakes will go away. That's a strong and compelling argument in the favor of vaccine. The mistakes that happen that confer additional viral fitness, true to what Darwin taught us, those will persist. Those that weaken the virus become extinct. And the US government developed a system of classifying variants that looks at each variant and evaluates, is it more infectious? Does it cause a more severe disease? And does it evade the current vaccinations that we have available to us? And based on those qualities of the variant, they came up with a classification system, including a variant of interest, which is the lowest concern, a variant of concern, sort of mid-level, and a variant of high consequence, which is, of course, the highest consequence. And fortunately, there are no variants of high consequence circulating in the US right now. And if you look mostly at the left side here, you can see this B117 is the variant that is growing in the US. That was originally from the UK, and it is a variant of concern. 35% of 80 viral isolates at Penn Medicine back in March were variants. So we are definitely dealing with a situation in the US and in Philadelphia where variants are becoming increasingly present. But here's the good news. If we look at vaccine efficacy, the fortunate thing is most of these vaccines induce an immune response that's 10 times or more than 10 times the presumed required level to prevent infection. Some of the variants cause about a 4 times reduction in sensitivity to the vaccines we have, but that still leaves us a really good cushion or a protective margin, as you can see in the graph that's here. So at this point in time, we're in a situation where the variants that are circulating remain sensitive enough to the vaccines that we have circulating. So that is not yet a concern, although it is absolutely possible it could become one. All right, so what happens next? What do we need to do next? It's important that we determine what the correlates of protection are, how much immunity is required from a vaccine to actually provide protection from infection or disease. We need to continue to collect additional safety and efficacy data in younger kids. We absolutely need to increase vaccine uptake and decrease vaccine hesitancy. We need to explore the role of passive immunization. And when I say passive immunization, what I mean is monoclonal antibodies. So both Lilly and Regeneron have developed monoclonal antibodies against SARS-CoV-2 that are highly effective as prevention. And what if in those who are significantly immune suppressed, who can't mount a really nice antibody response, what if they had an infusion of those antibodies every couple of months until we get to the end of the pandemic? Might that be a different strategy for protecting them from infection and disease? Obviously, we need to closely watch development and expansion of the viral variants in circulation. We definitely need to determine what the durability of the immune response from these vaccines are. So we continue to follow people on these studies out to 2 years. Even though we've determined that they're safe and efficacious, we want to continue to see how long does that immune response last, and do we need to provide a booster, either the same product delivered a third time, or do we need to tweak the vaccine for whatever variants are circulating? So looking at boosters, in the near future. And of course, we want to collect long-term safety data, although I will tell you, there is no history of serious adverse event associated with a vaccine emerging 2 months or more than 2 months out from the actual vaccination. So I think long-term safety data is likely to be quite safe. So I hope that you all feel armed with the facts and with some information that will help you to contradict many of the myths and disinformation that's out there. As nurses, we are amongst the most trusted professionals in the US. Poll after poll demonstrates that. And when people have polled the public to ask, who do you trust to get COVID-19 information and information about the vaccines that are circulating? And the number one answer in every single one of these polls has been people trust their own doctors and nurses, people they have a relationship with, who they already have developed a sense of trust with. Those are the folks whose recommendations they plan to follow most closely. And so each of us is in a really unique position to influence our patients, our colleagues, our family, our friends' decisions about COVID vaccine 19 uptake. So I know you're already out there changing the world, but I'm going to add a task to your list. And I just want to quickly say, it takes a village. None of this happens. None of this happens by one person or even a small team. So I just want to thank my colleagues in the clinic, Eileen, Nikki, Ian, Dave, Gail Broder at the COVID Prevention Network shared some of her slides with me that I could share with you today. All of the staff, and especially the Penn School of Nursing students who joined our team this year, making this work possible. And then perhaps most importantly, all those folks who came in and took a chance and rolled up their sleeves and said they wanted to make a difference in the world. So thanks so much.
Thank you, Deb, for such an amazing, I'm sure we're all applauding you, an amazing presentation. There are some questions in the chat I wanted to present to you. One question was from someone asking about what studies are being done to continue to monitor the original study participants.
So the original study participants are continuing in their original studies. We're going to follow them out to, I think one study is 18 months, one study is 24 months, so that we can be sure, long term that the immunity is durable and that there's no safety signals. Great.
Another question, is there information on how long the immunity lasts? You may have addressed some of this.
Yeah, we still need to learn that, because it hasn't been around for a long time. We just don't know that yet, but that's information we're still gathering.
And another question is, what is the comparison of vaccinations given versus presentation of myocarditis in teens presenting from the mRNA vaccine?
I'm not sure. What are the teens
so
presenting?
What is the comparison of vaccinations for myocarditis, the presentation of myocarditis in teens presenting with the mRNA vaccine? So it would just be the Moderna, Pfizer. What is the comparison for myocarditis?
I think we don't know yet. This is brand new. This has just happened in the last 2 weeks. The CDC has not published numbers that I'm aware of, and so I think all of that remains to be seen.
And another question was what about the safety of vaccines of taking lithium for bipolar disorder? This had someone tell their doctor was hesitant for him to get the vaccine.
Yeah, there's no reason to be hesitant. We included people with psychiatric illnesses in the trials, and there was no signal that any of the psychiatric medications interacted with the vaccine in any way that was harmful.
Great. Another question is, anyone currently giving antibodies to those who are immune compromised?
Not that I'm aware of, but it would not surprise me to see that be used as an off-label indication. Like I said, Lilly did a study in nursing home patients where their monoclonal antibody was very highly effective in preventing cases in nursing home residents and staff where an outbreak had started. Regeneron did the same thing in household members of people with disease. Also, it really worked well. So it won't surprise me to see us move in that direction.
And last question is about the issue of patients thinking that a magnet will adhere to the injection site. Have you heard that?
No, that's a new one to me. Don't think it's true. And I hadn't heard that.
Yeah, there are all types of experiences that are being promulgated and especially misinformation on Facebook and some of the social media. But thanks for sharing that. That'll give us an alert.
Yeah. I mean, we all have to remember we live in a time of amazing disinformation, right? I can't remember ever having as much disinformation about as important a topic circulated and believed as widely as exists for this. And so we have to be really careful when anybody tells us anything. What's the biologic plausibility of this being true? And to try to help people think that through.
And I want you all to go to the chat. Patricia Griffith has in there the program survey link. Please copy that link right now. You're going to go to that link and fill out the evaluation, and then we will then send you by email 2 to 3 weeks, give us some time, your CE certificate. Again, to reiterate, we are only giving CE certificates for this virtual attendance at this virtual live program. We will also be posting the recording that will have the slide on our website. If you go to the Penn Nursing website, there is a practice tab, and under the practice tab, there is our practice site. And in the practice site, there is a section on the right-hand side, a tab for preceptors. That's our preceptor site, and we will be posting the recording there. Give us a couple of days to do that. But the slides will be incorporated into the recording. So if there are no other questions, I want to thank you all again for attending this session, and I want to thank you, Deb, for just really invigorating us on accuracy and the science of vaccines. I know that I always learn something from you every encounter, and I'm sure that our participants did. And we just appreciate all of the work that you did to make this happen for our region and get this vaccine approved. I know that you worked and your team extremely hard in the months of when we were severely affected by COVID. And I want to thank all of you who attended, and those of you who are preceptors. Again, our many thanks, our sincere thanks for all that you do and making our educational mission, our teaching mission possible. And I'd also like to thank those of you who attended who aren't our preceptors. And if you're interested in being one of our preceptors, you can certainly contact us. Again, thank you all, and we appreciate you being here today.
Thank you.